Hello and welcome to this webinar using an automated CE-SDS platform in efficient biosimilar comparability studies. I'm Lou Rahman, Editor-in-Chief at DDW. I'm delighted to say that today's webinar will be presented by Dr. Richard L. Easton, Technical Director, Structural Analysis, BioPharmaSpec. The route to market for biosimilars places strong emphasis on the analytical and characterization techniques used to investigate the structure of the molecule and, most importantly, side-by-side comparability exercises between the biosimilar and innovator products. Both the EMA and FDA have set out clear guidelines for companies aiming to produce biosimilars, including the expectations for analytical investigation into structure. In these documents, the EMA and FDA both cite ICH Q6B as the guideline for biosimilar testing and highlight the need for using orthogonal techniques that enable cross-verification of data and conclusions from different methodologies. The UK MHRA has also published its own guidance document on biosimilars, with great emphasis placed on analytical data and a reconsideration of the requirement for comparative efficacy trials. Electrophoretic data forms part of the full structural characterization as given in the ICH Q6B guidelines, as well as serving to provide orthogonal data to other structural investigations, such as those using mass spectrometry. In this webinar, Dr. Easton discusses the use of automated Maurice platform to provide CE-SDS data for biosimilar comparability studies. First, some background on Richard. He obtained his PhD in glycoprotein structural characterization using mass spectrometry from Imperial College of Science, Technology and Medicine. He subsequently spent several years there as a postdoctoral research scientist involved in the detailed structural analysis of glycoproteins derived from systems expressing unusual glycosylation profiles. From this research, he moved to GlaxoSmithKline as head of mass spectrometry. Easton joined MScan, now part of SGF Life Sciences, in 2003 as a biochemist and became the team leader for carbohydrate analysis before being appointed principal scientist. In 2016, Easton joined BioPharmaSpec as technical director for structural analysis and is responsible for management of all aspects of carbohydrate and glycoprotein characterization. Thanks for joining us today, Richard. I'll hand over to you now for the presentation.
Hello everybody, thanks for joining me today for this webinar. I'm going to be talking about using the Maurice automated CE-SDS platform in efficient biosimilar comparability studies. Just to give you a little bit of background about the company that I work for, BioPharmaSpec is a contract research organisation that specialises in structural and biophysical characterisation services. We work really in the preclinical early phase R&D area. And we use state-of-the-art mass spectrometric and chromatographic instrumentation, along with a suite of other instruments, to provide full characterization, structural, analytical services that are needed within that area. We have labs in the UK. Our UK lab is based in Jersey in the Channel Islands, and a lab in the US as well in Pennsylvania. The company itself, BioPharmaSpec, is only 7 years old. But we came out of a previous company called MScan, which has a much longer history of research in this area of structural characterization. So a lot of our scientists have several decades of experience in terms of this sort of analytical work, and we can draw on that very extensively for the studies that we do. We have scientists who've been very heavily involved and indeed developed a lot of the techniques that are mainstays of the analytical arena as we use it now, such as peptide mapping and disulfide bridge analysis, as well as conceptualizing some of the main mass spectrometric instrumentation, such as the QTOF style of instrument. So back to biosimilars. Biosimilar analysis, as is, I guess, pretty well understood, very heavily relies on a lot of structural information. So if you are producing a biosimilar, you want to get your biosimilar to market, you have to have a lot of structural information to show that your product is the same as the reference product. And that requires very in-depth structural characterization in terms of comparability studies between multiple batches of your product against multiple batches of the innovator. And if you look at the biosimilar guidelines that all the regulatory authorities have put out, you'll find that this is very heavily and very clearly written within those documents. In each case, the regulatory authorities point back to this particular document here, the so-called ICH Q6B document. ICH stands for the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, so you can understand why they abbreviated that. The document itself has been around for a number of years now and goes into very clear detail about the types of structural characterization and the areas that we need to look at when we are going to fully characterize these products. So it defines the requirements at the primary structural level, such as amino acid analysis, peptide mapping, the glycan analysis, as well as more physicochemical type properties, the sort of gross properties of the molecules. So this will be the overall molecular weight, charge profiles, and in amongst there is electrophoretic pattern. So it's very clearly expected and understood that electrophoresis and generating electrophoretic profiles of these molecules forms part of a robust, expected and substantial characterization package. Over and above that, if you look at the regulatory authority guidelines, the other thing that they are very keen on is this idea of orthogonality, and all of the guidelines talk about this. Now, orthogonality in these terms is simply the idea that the data from one type of analytical procedure will support the data from another type of analytical procedure. So you have this kind of cross-verification of facts, of conclusions that are drawn from a particular procedure. And this idea of orthogonality makes for a very strong, very robust, self-supporting data package. And the sum is greater than the parts that make it up. So just to highlight this here, here's the FDA biosimilars guideline. And I've just pulled out one particular part where they talk about orthogonality. In this case, they're talking about it for assessment of aggregation, but they do use the term throughout the document on numerous occasions. So the idea really runs like a thread through the whole thing. If we look at the European Medicines Agency, they do a similar thing. And again, I've lifted a little bit of text here to show you. They talk about biosimilar comparability exercises using state-of-the-art instruments, state-of-the-art techniques, and again, orthogonal methods are mentioned there. They also make it clear that there is an expectation that these orthogonal methods will be used to look at product-related substances and product-related impurities. These could be post-translational modifications or indeed fragments of the molecules as well. The MHRA, the UK regulatory authority, as you can imagine, has followed with this idea. They talk about state-of-the-art instrumentation, state-of-the-art methods, and orthogonal procedures as well to look at similarities and to highlight, of course, any differences that may exist in the products. They take this one step further and they say that depending on the data that is generated, it may be that a comparative efficacy trial isn't necessarily needed. So in this case, products can be brought to market even more rapidly, but obviously the onus is on the data being of sufficient quality and sufficiently strong that a decision could be taken to not carry out that stage. So obviously there is a lot of requirement for strong analytical data out of all of this. We have responded to that by purchasing a Maurice CE-SDS system in 2020 in order to enhance our service offerings in this area. We have used it in numerous different projects across a range of different types of molecules, and I'm going to provide some of that information today so you can see how the system handles these various different types of molecules and the way that we've employed this in some of the analyses that we've performed. So the system itself is quite straightforward to use, which is obviously something that is really convenient. This runs right way through from instrument startup through to loading reagents and samples, which can be done in either relatively small vials, 2 or more vials, or 96-well plates can be used, through to inserting and running the cartridge, which again is a very straightforward thing. So the system itself is very convenient and very easy to use. But why would you want to use CE-SDS over slab gels if you have them? Well, there's a variety of reasons that this kind of more up-to-date procedure is more useful, I would say. CE-SDS has much shorter run times than traditional slab gels. You can run these in about half an hour. So obviously your throughput is that much higher. The The fact that you have automation of the runs means that you minimize your preparation time. So again, things have a higher throughput. UV detection of the sample, a direct UV detection of the sample rather than through dyeing of a slab gel, means that the relative quantitation is quick and quite accurate, which is obviously what we need for batch-to-batch comparative purposes and biosimilar to innovator purposes. That degree of accuracy of data that is produced is critical to allow you to confidently compare batches and identify any differences that may exist between the batches. For the Maurice system, it's also possible to use on-the-fly modification of batch parameters. So this means that during the course of a run, you can use fine-tuning to maybe have a particular set of characteristics that you require for a single batch in that run. So all in all, we find the system quick, convenient, and easy to use, and allows us to get through a good number of samples in a run. This is an example of the data, the type of data that we generate from the system. And I will come back to this image later to talk about it in a bit more detail. But as you can see, it's generated as a trace, an electropherogram. It's plotted in terms of relative migration time against absorbance. So the absorbance is the measurement of the amount of protein that's present in the bands. The peak areas that you will get as a result of the amount of UV absorbance are generated automatically and will be tabulated out in the data. But you can also manually assign peak labels, as we have done here, which allow easy identification of what those components or what those components potentially are as well. Now, as well as using relative migration time for this, you can also run a molecular weight ladder. And using the known molecular weights, you can translate this data from relative migration time to molecular weight as well, which is a very convenient thing, of course, to do. If you prefer looking at things in more traditional ways, then it is also possible within the software, the Compass for ICE software, to take the data that you would normally have in the electropherogram profile and visualize that in the so-called virtual gel format. And the data that you see here is the same data that was on the previous slide, but now presented in more of a traditional kind of way. So if you find it easier to visualize information like this, then obviously you have that capability there. Then it may be that taking this data and comparing to other data that you have from other systems is more convenient in this form. So what I'd like to do now is just talk about 3 different types of molecule that we have analyzed with this system, and just give you an example of some of the data that we've generated and the various different analyses and methodologies that we've applied to these samples and how those have run through the Maurice system. So first of all, if we look at monoclonal antibodies, now it pretty much goes without saying that this is a massive area within the biosimilar industry. There's a huge number of products that are out there and many more that are on the way that are monoclonal antibodies. Monoclonal antibodies themselves, I'm talking about IgGs here, I should say, are tetrameric species, tetrameric molecules, composed of 2 light chains and 2 heavy chains. So you could almost consider them as a dimer of dimers, if you like. These tetrameric molecules, these 2 heavy chains and these 2 light chains, held together by 16 disulfide bridges. Now, this means that it's quite a complicated structure. You are looking at a fairly complex process to produce these intact species. And sometimes things go a bit awry. So as well as producing the intact molecule, you may well also be producing in your product partial species. So it may be that rather than having a tetrameric structure, you have a population of dimeric structures, maybe a heavy chain and a light chain, possibly 3 light chains, possibly 3 chains bound together, as well as the vast majority in the correct tetrameric form. Now, you can assess for this very easily using the Maurice system. It gives very clear separation of monoclonal antibodies and any partial species that may be present. And this data here represents 6 batches of denosumab, which is used to treat bone loss. And in it, you can see that we have a large amount of the intact monoclonal antibody, as you would expect, but also there are smaller amounts of other species present as well. And they've been labeled here as light chain, a heavy light chain dimer, a heavy heavy chain dimer, and a heavy heavy light chain trimer. Now, if we want to confirm the components that make up these extra peaks, and we've got a fair idea of what they're going to be based on the masses of heavy and light chain and the summation of those masses to give these profiles. We can take this sample and we can carry out a reduction on it. So now these samples have gone from mixtures of heavy light and light chain to free heavy and light chain species as a result of the reduction. The disulfide bridges that held these chains together have been broken and all chains are now free. So if you look at the right-hand image at the top, and I should say it's important to note that the scales are different here. So the large peak you see on the right-hand side of the right-hand image is not the intact monoclonal antibody now, but is in fact free heavy chain. Okay, so this is our 6 batches showing free heavy chain and also free light chain. And those other components that we were seeing, the dimers, trimers, and the monomeric light chain species is still there because it's now been released. But the other dimer and trimer components are no longer present as a result of the reduction of these samples. You can also see just to the left of the heavy chain is another small component, and that is potentially a non-glycosylated heavy chain. It's the heavy chain of a monoclonal antibody that carries the glycosylation, and there could be a small amount that has not been glycosylated during the biosynthesis of this molecule. Now, in order to confirm that, we can take these reduced components and we can carry out the de-N-glycosylation using the enzyme PEGylated. Now, what you see here on the left is just 3 stacked batches of the denosumab that have been reduced. So basically it's 3 runs from the previous data, but now shown stacked. And in each case, we treat those reduced samples with the enzyme PEGylated. And you can see that now running those enzyme treated samples, the heavy chain peak is significantly reduced, but the peak at the position of the non-glycosylated heavy chain is now significantly increased as a result of the conversion of the heavy chain to its non-glycosylated form. And this is running at exactly the same position as the peak that we were seeing originally in the reduced non-glycosylated heavy chain, confirming that as the identity. The PEGylated hasn't worked to 100% completion, which is why we still have some glycosylated heavy chain present. So we can use these various different biochemical and enzymic procedures to probe the structure and the peaks that we're seeing from the CE-SDS sample and confirm their identities. So this is a very powerful way of assessing your batches, your molecules, in a rapid and quite automated way. So the next molecule I'd like to look at is a molecule called etanercept. Now, etanercept is a very heavily glycosylated molecule. I mentioned just now that monoclonal antibodies are glycosylated, and indeed they are, but the amount of glycan in a monoclonal antibody is only about 1% by mass. So relatively speaking, it's a small amount of glycosylation for the large amount of protein that's present. Etanercept is very different, and this is true of quite a lot of the molecules that we work on, quite a lot of the glycoproteins. They are more heavily glycosylated than monoclonal antibodies. Etanercept is particularly heavily glycosylated. So the molecule itself is a fusion protein of the TNF receptor extracellular domain with the IgG1 Fc region. It's a dimer, and it contains 6 N-glycosylation sites and up to 28 O-glycosylation sites, although there is some degree of variability in terms of the amount of O-glycosylation that you will see in the population. Now, if we take this etanercept and we run it by CE-SDS, this is what we see. If we run it non-reduced, we get a single peak that is relatively broad at high mass. Now, the mass, as determined by the system, is written at the top, 206 kilodaltons. This is much higher than the actual mass of etanercept. The reason for that is the very large amount of glycosylation has a very large hydrodynamic volume. It has a lot of negative charges taking up a lot of space, and you will get an anomalously high run position. Nonetheless, we are using the Maurice system really as a batch-to-batch comparative tool and not as a mechanism of getting an accurate mass on it per se. So it's perfectly fine that we see a higher mass than we would expect, in comparative terms, the 3 batches that have been run here are all showing the same thing, and that's the important thing. They're all handling in the same way. Now, that peak is quite broad because the glycosylation will have a high degree of heterogeneity. There will be variation in fucosylation, there will be variation in silylation, there will be variation in the degree of lactosamine repeats that may be present. And all of this adds up across the population of molecules to produce a very heterogeneous mixture, and therefore quite a broad peak. If we take that sample and we reduce it down to its monomeric forms, you can see we get a signal, as you'd expect, at half the mass, 103 kilodaltons now. And it's a narrower peak because we are decreasing the heterogeneity. We can also see that in the upper trace, there is an absence of monomeric signal because we now know the run position from the reduction, and we can see quite clearly that there's nothing there. So in these samples, we have no monomer and 100% dimer, which is obviously good to know. Now, if we take these samples and we reduce the heterogeneity by de-N-glycosylating with PNJZF, just as we did for monoclonal antibodies, as you can see, we get a decrease in the mass, and we're also starting to narrow down the width of the peak by decreasing the heterogeneity through removal of the N-glycans. Now, you can also see we're starting to get some variation in the peak profiles between the batches. And this is really useful because we can start to use this to see how different batches are handling and develop range information, if you like, between biosimilar batches and innovator batches, and see the degree of overlap in terms of the profiles that you get between the biosimilars and the innovators. So we're starting to tease structure out of this through the use of this digestion. Again, we can take this one step further and perform a reduction on the molecule, and this helps to decrease that heterogeneity further by splitting the molecule down into its monomeric forms. And you can see that in the lower right-hand corner of the chase, as I'm showing here. This is now showing a clear signal at 76 kilodaltons. And again, you can see some definition between the peaks that are run there. Now, it's important to remember, of course, that we've de-N-glycosylated, but we haven't de-O-glycosylated. So the O-glycans are still present. Now, there is no clear or easy way to enzymically remove O-glycans from a molecule. So in this case, we've reduced the heterogeneity by, rather than removing the O-glycans, by using an enzyme to desilylate the O-glycans that are present. So again, we are reducing the heterogeneity by removing one of the sources of heterogeneity from the remaining O-glycans. And again, you can start to see that we're getting more peak definition from the non-reduced sample. The mass again has decreased, as you would expect, because we've removed the sialic acids. And we can start to see some structure in that main peak there. And it looks like some of the molecules are partially de-N-glycosylated, but not completely de-N-glycosylated, as well as showing some variance in terms of the overall peak profile as well. Again, we can take this sample and we can monomerize it. And again, the peak becomes that much tighter because we've reduced the heterogeneity still further. And the definition around that peak is now becoming more clearly resolved. So we can use a variety of procedures, in this case, enzymic procedures focused around the glycosylation, to start to tease out various different structural aspects of the sample as we see it. Now, I've talked about orthogonality, and obviously this is important in structural characterization work, particularly for biosimilars. So I thought it'd be interesting just to show how the CE-SDS data aligns with some data from mass spectrometry. So if we take our non-reduced sample here, so this is just the same data as you saw on the previous slide with our signal at 206 daltons by CE-SDS. If we analyze this sample by matrix-assisted laser desorption ionization, we get 2 peaks. Now, the peak at 121,179 is the mass of the dimeric molecules. That's 121 kilodaltons. The peak at about half that mass, 60 kilodaltons, could either be the monomeric form, or because of the manner in which data is produced by mass spectrometry, it could actually be the doubly charged form of the dimer. Okay, so it's not necessarily the monomer. Now, we know that this isn't monomeric data because according to the CES data, we have no monomer because we know where the monomer runs, and we don't see that. So we can be confident that what we're seeing is not monomeric data by MALDI, but is in fact a doubly charged dimer. So we can confidently assign that component. If we take our reduced data and we run that by mass spectrometry, we get the peak that we would expect at the mass of 60 kilodaltons. In this case, we have it because we've actually produced the monomeric species rather than knowing that it could be possibly the doubly charged form. So this is an example of how different techniques can support one another and how we can use data from one to affirm conclusions that we're drawing from another. The other thing I thought it'd be interesting to show is a couple of examples of electrophoretic data. Now, in this case, this is the etanercept, as you saw before. This is the de-glycosylated data compared to the desalylated deglycosylated data, both of them reduced. And you can see the differences that have taken place there, the sharpness of the peak and the decrease in mass. Now, if we take this data and we compare it to etanercept run natively, which is in blue, and following desalylation in red, by imaged capillary isoelectric focusing, we're now looking at the difference in these samples based on their charge profiles rather than based on their differences in mass. So you can see here that desalylated data in red is now far less acidic, as you would expect. We can look at the PI scale across the bottom, and we can see that on the left we have signals around about 4.5 to 5.5 PI units, whereas following desalylation we're slightly more basic, we're just 7 to 8. We've also decreased the heterogeneity a little as well, because we have taken the source of that heterogeneity away, the variation in the charges based on the sialic acid. Now, there will be other factors at play here in terms of defining precise charge profiles, so that's why you don't have a single peak for the desalylated species. But you can see that they are now considerably more basic and less heterogeneous as a result of the treatment with the sialidase. So the final thing I just wanted to touch on is a modification to molecules that is very much man-made, and this is the attachment of polyethylene glycol units, so-called PEGylation. Now, this is quite a common modification for biopharmaceuticals. It serves a variety of purposes, such as increasing the drug half-life, changing the protein solubility properties, all of these designed to minimize patient requirements for drugs or discomfort through taking drugs, to increase the longevity of the product in the body. So CE-SDS is a very useful tool for evaluating PEGylation. These units can be quite large, anywhere between 5, 20 or more kilodaltons added onto the molecule. So we can use CE-SDS to look at PEGylated and non-PEGylated species, and also to examine the extent of PEGylation within a molecule. And I've got an example of that here. So this represents 3 batches of a PEGylated protein. All of these are running at 33 kilodaltons, and you can see that they overlay very nicely indeed. So there's good consistency here, there's good process control, and there's good reproducibility of the PEGylation method. But if you look carefully, you can see there is a small signal at 52 kilodaltons. Now, this is consistent with over-PEGylation. So there's a small percentage of the molecule, anywhere between 0.3 and 0.5 in these batches, where an extra PEG molecule has been added. Now, knowing this, the site or potential site of PEGylation can be investigated through other techniques, such as mass spectrometric assessment of the mass overall, peptide mapping of the molecule to identify where this is attached, site investigations using chemical labels to find where the PEG was attached. All of these can be carried out specifically targeting or being used to specifically identify where this over-PEGylation is taking place, if indeed there is a specific site. It's then a case of going back, potentially readjusting the process to eliminate that over-PEGylation if it's an issue. So in summary, what I really wanted to point out is that biosimilar studies do require orthogonal data across all areas of structural analysis. There is an expectation that this will be performed, and that by doing so, you will generate a robust data package that you can then put forward to support any claim of biosimilarity. Now, we have used the Maurice CE-SDS system in numerous structural investigations in the area of biosimilarity, and we found that we can get very key data on a variety of different molecular types using this system. Monoclonal antibodies, heavily glycosylated proteins, and PEGylated proteins, amongst other things, have all been investigated, and they've all been shown to work well in the system, and the system is able to, along with appropriate chemical and biochemical means, allow us to tease out structural information using this system. We found that it's very reproducible, very easy to use, and it does allow us to very confidently perform batch-to-batch comparative investigations for various clients across various different projects. But I would also like to thank Lyndon Gledhill and Michael John O'Connor for doing this work. I should say they are the people who produced the lab work. I've presented it to support them, but it's very much their work. So yeah, a very big thank you to them for their efforts in producing all of that data. If you do have any questions, then please feel free to get in touch with me, or you can speak to my colleagues in the business development team. We cover all areas globally, US, UK, Asia, and India. So hopefully you can find someone in your time zone that you can speak to directly. And also, please do look at our website for more details. We have a lot of information on there about this technique as well as other techniques. So please feel free to take a look. And if I can help in any way, please let me know. Thank you for your attention.
Thank you, Richard. That was an excellent presentation there. Before we continue to questions, I'd just like to remind everyone that this webinar will be available to access on demand on the DDW website, www.ddw-online.com. We're now going to pause for a few moments to allow audience members to send through any questions that they might have for Richard. Okay, so we'll now go over to questions if you're ready, Richard.
Yes, that's fine, yes.
Okay, so the first one that we have is, can you elaborate on what you mean by on-the-fly modifications?
Yeah, sure. So when you begin a batch on the Maurice system, you can add pauses after certain samples. So what this means is that you can quickly review data that you've generated up to that point, and then you can add new runs with modified parameters to fine-tune your data. So this is useful as all injections remain in one batch and reagents don't get used too much, but it does mean that you can make any tweaks that may be necessary as a result of what you're seeing. So it's a very useful way of being able to modify things as samples are running.
Okay, thank you. You mentioned loading samples and reagents, but what does this actually entail?
Yes, so all reagents, they come ready to use from Protein Simple, or at least all reagents that are associated with the Maurice system. So it's just a case of transferring reagents from a bottle to a vial. So it's very quick and very simple, and does give you high reproducibility because you can obviously have faith in the reagents, and it means that things are very quick and the automation process is very quick to load, to set the system up, to get everything operational and running.
Okay, and what about the cartridge, Richard? Does this require any maintenance or preparation?
Well, the cartridges do come ready to use, and they're good for 500 injections or 25 batches. All that's required in terms of preparation really is to insert the top running buffer into the cartridge before putting the cartridge into the system. Cartridge cleaning does occur at the end of each batch, and the cartridges can be stored until they're needed again. That is really all that you need to do.
Okay, thank you. And the next question that we have is, what is Compass for ICE like to use?
It's a fairly intuitive piece of software, actually. It's used for both controlling the instrument and processing the data. Building a sample list is pretty simple. You just specify the sample location, tell the instrument which methods you want to use on which sample. The processing itself was also streamlined. Most peaks are automatically detected. For the weaker peaks, detection thresholds can be tweaked. And usually it's just a case of selecting those peaks, adding it into the software, and then the software does the rest. Extra features such as peak naming and generating these virtual gels is also really useful, as you saw earlier.
Thanks, Richard. Another listener has asked, there's a peak labelled IS on all of the traces. What is this?
Oh, yes. So IS is the internal standard, and apologies for not pointing this out. The internal standard is added into each sample and is a 10 kilodalton marker protein that's used to normalize the relative migration time. So that is the x-axis that you'd see on the electropherogram. So the run position of the internal standard is defined as one relative migration time, and the other migration times are set up relative to that.
And the next question is, how are you getting molecular weight information from relative migration time?
Right, okay, so if you want to translate that into molecular weight information, you can run a molecular weight ladder within the batch. This obviously contains peaks of no molecular weights. So you can then tell the software which peaks of which molecular weight running at which relative migration time. The software can then convert that migration time into molecular weight based on that information. So effectively, it's sort of like a calibration.
Okay, and then finally, Richard, the last question that we've had in is CE-SDS a technique that you find you're frequently being asked for?
Yes, it is. We use this technique in quite a variety of different studies. In some instances, we have clients who specifically want us just to perform CE-SDS for them. In other instances, we are doing, as I mentioned earlier, these much fuller, in-depth biosimilar comparability studies. And then most certainly we put it in there. As you saw before, it's a requirement of the ICH Q6B guidelines that you generate these electrophoretic profiles. But at the same time, using this system and using some of the procedures that we've talked about in terms of the biochemical manipulations on the samples means that we can generate really useful, meaningful data that stands alongside data generated from other techniques and helps to produce this really robust package. So yes, most definitely we're using it a lot, and it's really quite a key part of our services.
Thanks for those responses there, Richard, as well as such an informative presentation. If you'd like to listen to the webinar again, it will be available on demand at www.ddw-online.com. And you can now view Drug Discovery World content online on your mobile or tablet or via the new DDW app, which can be downloaded via the App Store or Google Play. Thank you for listening.